Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Richard Jacobs with the Future Tech and Finding Genius Podcast. I have Gaël McGill. He's the president and CEO of Digizyme. He's the director of molecular visualization in the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. So, Gaël, thanks for coming. How are you doing?
Good. I'm great. Thanks for having me on the podcast.
What's your research about and your work at Digizyme? How do the two coincide?
So I'm mostly interested in how we visualize science, how we understand it through images. And as we'll discuss, we tend to approach that in a number of different ways. So not only through projects in the real world with companies and schools and museums and things like that, but we're also interested in the design decisions that lead to effective imagery for communicating science. And that's more the side that I try to tackle as an academic and with kind of federally funded research at Harvard Medical School. So it's really...
When you talk about imagery, do you talk about images used in the clinic, like MRI images, or do you talk about teaching science and showing examples of stuff?
Yeah, no, that's a great point. So the imagery that we usually create is not the imagery that is coming straight out of either medical or other instruments. So that's, of course, not to say about the relative importance of those images as well. It goes without saying. But we tend to create imagery that is for teaching, but that also, I think of it as knowledge synthesis. So combining a lot of different data into representations that help you better understand complex science. And also representing areas of science. I mean, I'm a molecular biologist by training, in areas where there is no microscope powerful enough, frankly, to actually get a direct image. So there are some realms of science, like the molecular cellular realm, where we actually need software to create imagery of molecules because they're too small to see with microscopes.
So if I see a really cool visualization of how DNA gets transcribed, it might have come from you, like on YouTube and stuff, right?
It's quite possible. I thought you were going to say it was probably coming out of a software program as opposed to a microscope, and the answer would have been yes. In other words, it's one of the things I'm interested in, which is how people respond or understand when they're looking at images that are right out of instruments versus someone had to create this in order for you to see it. And the minute that someone has to create it, and the minute that it's created from data, it kind of creates a whole host of questions about how and why it was created, what is the kind of artistic license that was used. And that's where, again, I was saying that this aspect of how do we design imagery for it to be most effective in teaching and communication, that's kind of another aspect that I'm very interested in. So
I think people love these visualizations. I mean, I know I do. I crave them. I always want to know what something looks like. And, if they're done right, it can really advance your knowledge of a given area. Like, there's this contest, I think it's by Nikon, or I don't know which camera company, but, it's at least light microscopy, what could be seen, and, the most amazing imagery there. And that's super informative. But, the stuff you do as well is. So, yeah, that's my feeling about it.
No, it's like, as a scientist, it's like being a kid in a candy store. I mean, it's an endless series of topics, even beyond one's immediate field of expertise. So again, even though I'm a cell molecular biologist, I've probably learned more science as my role in leading Digizyme as a science visualization company than in the 6 years of my PhD, where I focus on one little topic. Scientific visualization reaches all the way from, again, the atomic to geological evolutionary timescales and everything in between. So it's really, yes, the result is images, and that's why we call it visualization or animations. But I think it's the process that we go through in order to create them that's actually most interesting, and is maybe kind of the hidden part of the iceberg for most people. They just see the result of our work. I think what's very powerful is all of the information and data that you need to synthesize in order to create an effective visual. And that's why it's such an exciting thing to do on a daily basis.
Yeah, like I wish they would have I looked at anatomy books and that's static, but I wish they would have a 3D program where you could look at various parts of the body and, have the body sit or lay down or stand or run, and you could zoom in and pan and look under and around things and take things apart and strip off layers. And, even the human body, I don't know of any software that does that. Like I've seen in movies, someone kicks someone and crushes their skull supposedly, and they make this like little animation of it. But I know that has nothing to do with reality. I wish they would have software like this, for instance, about the human body. That would be really cool.
Yeah. Well, we're getting there. I think with the new technologies, not just 3D animation, but with things like VR and even AR, virtual reality, augmented reality, there are programs. And I should mention, if we are talking kind of at the human body anatomy level, there is one professional organization in particular called the AMI, the American Medical Illustrators Association, which is just a fantastic place to go, and an amazing group of individuals who are formally trained both in science and in medical sciences, as well as in art, and wielding a whole number of technologies, digital technologies, from handheld pencil to digital painting techniques, animation, and even VR. So it's through communities like that, and I think the efforts of those folks and ourselves. I mean, we're part of that community where we're trying to create more and more of these educational tools that kind of provide the best of both interactive technologies and what we know about effective pedagogy and design.
So, what about with things that you can't see? At the nanometer scale, inside of a cell, inside of a bacteria or a virus. I mean, how do you begin to make visualizations of those?
Yeah, that's actually, I'm interested in many things, but I confess I always have a sweet spot for the infinitely small, because that's where my passion for science really started. So yeah, I mean, as I was mentioning earlier, we obviously have very powerful instruments. There are microscopes that are based on light, so light microscopes. At some point, you reach kind of the lower limit of what the visible spectrum and those wavelengths of light can do, and you have to go to other methods like electron microscopy. But even then, as you go further and further down and try to see features that are smaller and smaller, you basically hit a limit. And that's where other techniques come into play, ones such as X-ray crystallography, or what a recent Nobel Prize was awarded in is this other technique called cryo-electron microscopy. So, but to make a long story short, these are techniques that essentially allow you to shine rays or beams through preparations, crystals in the case of X-ray, but even more disorganized preparations in the case of cryo-EM. And by analyzing how these beams are deflected, are bounced back, you can reconstitute an image and get the information for the precise location of every single atom in a molecule. And that molecule can be, small, like a hormone or something like that, or it can be rather large, like an entire virus. So we actually have really good data on how these things look like, and we're getting more and more of these data, but these are just data. And so at that point, you need to take what is essentially a 3D coordinate list and create an image from that. And that's basically where we come in and even run with that even further. In other words, not just static images of, let's say, what a virus looks like. And unfortunately, we've been seeing a lot of those recently in particular, but we're very interested when you have multiple snapshots of what the same virus or protein or molecule looks like in different states. You can also actually create morphs or animations of how these things might go from one state to another. So to make a long story short, software-based reconstructions can be very powerful at leveraging these data sets for very small things like molecules or even parts of cells.
You know, whenever I've spoken to someone that's in a particular field, they always hate when movies have been made about it or shows, because they're always like so horribly inaccurate. So when you see stuff on YouTube, animations and things like that, you know, are a lot of them accurate? I know this is like a very wide ranging question, or are a lot of them completely inaccurate? Are they good faith attempts to really see what's going on? Like what's, you know, if I just go on YouTube and look up stuff, am I likely to see accurate stuff or not?
Right. No, that's actually really, it is a huge question, but it's a great one I really like, because one of my favorite slides to use actually when I give seminar, just even scientific seminars at conferences or various places, I have one slide where I've collected at one point the top, if I just search DNA in, let's say, Google Images, I collect the top 10 images. And this may have changed because, of course, the search results shuffle around over time. But at one point, so I put all these pictures of DNA on a slide, and then I ask this scientific audience, I say, what do all these DNAs have in common? And usually, you know, there's kind of a silence that spreads over the audience as they inspect these pictures of molecules. And the answer is that they're all wrong in one way or another. So either they're lacking an important feature, which is this major minor groove. So it's not just a twisted ladder, but not to get into the weeds, but that's usually one issue. Another issue is that it's usually twisting the wrong way. You know, it's not a right-handed helix that's depicted as a left-handed helix. The reason I use that slide is to try to get people to, you know, it's hard to speak about just accurate or not accurate. I think that the way I rather get people to think about it is this kind of famous sentence of, you know, all models are wrong, some are useful. It really depends on the useful part. So we are rather obsessed with accuracy, I have to say, because everyone, unlike some of the other, I guess, groups that we work with, or perhaps you might say compete against in the field, pretty much everyone on the Digizyme team is a scientist by training. So one of the differences in the way that we've approached our work, and it's not to say that collaborations between scientists and artists or animators are not great. They are, and they've led to wonderful work. But in putting together my own team, we've found that to really do justice to the science and be able to make design decisions that don't oversimplify, it's been important to find people who really combine both kind of graduate PhD level understanding of the science and you know, production quality, you know, animation, graphic design capabilities. So because of these kind of dually trained backgrounds, that's not to say that we always get it right, but we are driven kind of by our passion for the underlying science. And so that's usually where we start. And we really try to understand what are the key features. And understanding the science doesn't necessarily make it easier to then make the right design decisions, which is a whole separate can of worms. In other words, some things are so complex to depict that in order to make it understandable, you still need to make all kinds of decisions for what you're going to leave out, because you can't show everything.
Oh, what's an example of that?
Oh my gosh. Well, pretty much any picture of a molecule that you've seen has probably had just to pick an immediate example, all of the surrounding molecular environment has most likely been removed for you. So I know that sounds really kind of simple, but just to start with the basics, we were just talking about pictures of DNA. If you're showing me a picture of a beautiful, even if the DNA itself is correct in its structure, but it's on kind of an empty background, by definition, that's a design decision right there, because DNA does not float around in air. It's usually in a solution. It could be in a test tube of water, and in that case, someone has decided to remove the depiction of all the water molecules around the DNA, but they're there, right? Or in the case of a cell, which is usually where people are depicting things, so they're trying to show you how a gene is getting turned on or something of the DNA is getting mutated. Those environments are unbelievably crowded and complex. And not only that, but they're moving at speeds that are completely outside the realm of the human senses or experience, right? So
Really? How fast? What do you mean?
we're talking picosecond, femtosecond. I mean, ranges that are orders and orders and orders of magnitude faster than anything we can imagine. It's
Wow.
the analogy in the other direction is if I tell you that, Precambrian fossils 600 million years ago, I can give you that number, but as a human, how do we intuit those numbers? We can't, right? We can learn them, we can memorize them, but they mean very little to us intuitively. The same thing happens. Yeah.
How do you even then know that you're, I don't know, how do you know that you're close to imaging the real thing? I mean, I've heard about the molecular storm, for instance, and I guess have you ever tried to create images like that? Like, here's the image we created for you, and here's what we guess it might really look like, no matter how messy.
So, but keep in mind that the look like is not necessarily the is. So when you ask, the first part of the question is, how do you know? We do have instruments, though, that give us data, and that data informs us on the size, the speed, all kinds of characteristics. Then there's the separate question of how do you depict that in imagery for kind of human consumption. And that's where, so to get to the second part of your question, how do we, have we ever attempted that? Not only we've attempted it, but it's actually the basis for some of the research now that I do kind of with my academic cap on. So as part of my work at Harvard Medical School, and I collaborate with wonderful researchers also at the University of Toronto, namely Jodie Jenkinson in her lab there. What we're trying to look at is when we create, for example, the same short molecular movie. So let's say we're looking at some hormone binding on the receptor on the surface of a cell. Short sequence, maybe 20 seconds. So nothing fancy. But we create that movie, let's say, in 4 different versions. We create a version that I would call the most kind of typical version, where the hormone, is flying through space in a very gentle arc. It gently slows down, it lands on the receptor. The receptor on the surface of the cell seems to know the hormone is coming, and, they embrace, and boom, that's your typical molecular animation. It almost uses kind of the Disney principles of animation, which is nice overlapping motion. The proteins know where they're going. Everything's nice and graceful, and nothing's around them. It's just them, the molecular actors. Well, that's version 1. Version 2 might be exactly the same actors, but now the path of both the receptor and that hormone is going absolutely nuts. In other words, it's a random walk, which it really is in real life. It is a random walk. So that's version 2. Version 3 might be that same thing as version 2 with messy motion on both receptor and ligand or hormone. But now we've added a host of other molecular actors that surround these 2 proteins. So it's getting messy visually. And then the version 4, which is typically my favorite, is the complete mess, where basically think of version 3, but now we've added all of the molecular water, the stuff that people almost never show. So why am I taking you through this? Well, we create 4 versions because then we can take, let's say, undergraduate students, hundreds of them, and we can break them up into 4 groups and give them like a pre-test and a post-test, and each group will see a different version of that same animation. And we can do eye tracking on the students so that we actually know what they are looking at during those 20 seconds. We can ask them to describe what they're seeing, so do kind of a concurrent verbalization. And then we can do a long-term delayed post-test 2 weeks later. And all of this is in hopes of understanding which is the version that helps students better learn some of the key features that you're trying for them to understand.
Why would it be one version? Why wouldn't you show the simplest and then more and more complex until it's approaching reality? I mean, this is what I learned in engineering.
Precisely. I mean, that's exactly what you want to do. And it seems so obvious, and yet it's not what's done. So typically what a teacher would do is to go, let's say you're teaching, I don't know, the cell cycle or some topic. Typically what they will do, and everything's a generalization, but as a general kind of approach to this, you go off and you look for the best possible cell cycle animation to show your students. And students love the animations. You usually get quite an attention bump. I think of it as almost like attention credits, right? You bring an animation to class, students will love it. But how often will teachers take 2 or 3 animations of the same topic? They don't have to be long, but multiple ones created by different people, and have the basis of the discussion not be about, what did we learn, but how are the versions different and what was left out in some of them? And do you think this one's right? Why not? Or why do you think about this one's right? So in other words, what I'm interested in pedagogically is involving people in thinking about how the images were made, because it turns out that in and of itself is a fantastic way to learn the material, right? I mean, science, not to get too philosophical here, but science is not a bunch of facts, right? Science is a way of knowing, and it has to be taught that way.
What would be nice too is if you have a system and you do models of it, and you say, this is assumption set 1, and here's the models we create, and here's now assumption set 2,
right,
and here's the model we create. I don't know if everything will be amenable to that, but that would also teach you even more about a certain function or a certain thing.
No, absolutely. And I mean, you're hitting with your questions on kind of, which is great, on different aspects of our work. So even though I just described kind of the kind of experiment that we run, again, kind of in my federally funded, part of my activities, what you've just described is also something we kind of published on pretty recently, which is this notion of, wouldn't it be nice if the visualizations came with some sort of metadata system that would indicate not only how they were made, but as you said, what are the, what hypothesis or what model is this depicting, and based on what data, and how does it compare to this other model? And we're trying to develop tools that let you basically take these movies and add, kind of an interactive, like HTML5 wrapper around them, and that let us annotate these visualizations so that people don't just see the result of the visualization, but they can gain more information about how they were made and what is the level of accuracy that was used.
Yeah. You must have, I don't know, since you're a visual guy, and in this regard, I am too. Like, I can tell you, I got Frank Netter's anatomy book. He's a medical illustrator, and I looked through it a bunch of times, and I said to myself, let's see what I learned. And I noticed just by looking at the images many times, all this information comes out at you that you never would have even suspected you'd figure out. So what have you figured out? What kind of things have you looked at where ideas came to you and you had to rush to tell somebody, like things that you figured out by looking, or even feedback from people that have looked, that have come up with new and really amazing insights?
I mean, it's literally why I get up every morning. I mean, every day, I won't say maybe every minute, but it's really, I mean, it is the core of our work. In other words, and it's not just that it's exciting for us who are doing it, but what I find constantly fascinating, and I'm trying to, in a sense, a little bit of my soapbox is to try to get the scientific community and even industry to gain a better appreciation for the benefits of visualization. Again, people tend to focus on the end product. Like, I want a pretty animation of our mechanism of action, or some, here's a new drug, we want to make an animation of how it works. But the number of times that we've worked with experts in their field, so literally some guy who is the world expert on, let's say, the proteins on the surface of HIV or some other virus, this is the person who's actually generated the data, right? So they've published, they're the ones who have known these proteins and know what they look like. When you engage these people in creating a visualization with them, you start to see these mini epiphanies that happen. And it's always surprising to me because, again, these are already the experts. And yet in the process of having to create a visual or a visual model, a dynamic model of their work, they're having to externalize some of their knowledge in ways that other activities like teaching or writing don't necessarily force them to do. So it's for me, so you asked for a specific example. I mean, right now we're in the midst of creating, this is kind of an in-house project, not driven by a client or a commission, but we're in the process of creating a full cell entry animation visualization for the SARS-CoV-2 virus. And I think it's actually a good case study for us because Lord knows we've seen a bunch of really beautiful images in the news. There's the image from the CDC. There's a lot of interesting activity in terms of people kind of creating pretty pictures of the virus, and not just pretty, based on available data that's been published, which is great. But what we haven't really seen yet, and this is what we want to try to contribute to the community, both for outreach, but also for the scientific community, there's an incredible choreography, a molecular choreography that happens from the time that the virus first binds cells that express the right receptor to the time that the virus can actually penetrate the cell's membrane and release its genetic payload.
Yeah, I've seen one animation where a virus turned itself inside out, and it was like a crane unfolding, and then that pulled the membrane apart and entered, and it was amazing.
Yeah, I mean that's exactly what happens. The big barrier for viruses is that cells are surrounded by these membranes, these kind of fatty-like layers of molecules made of lipids, and for most hydrophilic, in other words, for things like viruses or other molecules, it's very difficult to go through that membrane. That's part of the point of the membrane. So in order for the virus to begin infection, to get into your cell, it has to, in the case of SARS, it has to merge its membrane with the cell's membrane. But that's also very energetically expensive to do. In other words, from a kind of a thermodynamic perspective, like these things don't just happen. So to make a long story short, there's this complicated protein machinery that lives on those spikes on the surface of the coronavirus. Those are not only responsible for tethering the virus to the surface of its target cells, but once that virus is tethered, it has this incredible refolding of this protein that drives the membranes to fuse together. And once they do, it's game over in the sense that the virus is not able to enter the cell. So depicting those conformational changes, depicting how that viral protein is driving that process is something that we actually know a lot about. We have studies going back, 20 years in other very similar beta coronaviruses that we can use to assemble kind of a full visualization of that process. So just being engaged in summarizing that data and creating that visualization has been an incredible eye-opener for me, at least, to better understand how this thing works. And if you know how it works, you can, of course, better devise methods to inhibit it.
Well, in that regard specifically, I wonder if it's one virus entering all on its own, or if it's one, more than one coordinating, and that's how they get through the membrane. But that's, a detailed nuance that maybe you don't know about, or I don't know.
Well, no, I mean, we do. I mean, so without going too deep into just viruses in general, it's really interesting. So there's a lot of variety among viruses, right? From HIV, flu, SARS, dengue. I mean, the list goes on, and they're all devised these really. If you just look at their sequence and the shapes of their proteins and just what they look like, there's a great deal of variety. What's interesting, though, is that underneath that variety at first glance is very similar machines. In fact, most viruses can just be popped into these two categories for their membrane fusion machinery. So that's just another way of saying that if you study how flu enters cells, or if you study how HIV enters cells, you actually learn a lot about how SARS enters cells. And so these machineries have a lot in common. And I won't go into the details of it, but that's how we're able to make some pretty good educated guesses as to how, let's say, the new virus machinery works based on all the data that's been accumulated from these other viruses. And you're right, it probably takes more than just one spike protein. It probably takes the action of at least two of them to kind of refold and drive this membrane fusion.
Yeah, I wonder if it's like the Matrix, where those machines landed on the Nebuchadnezzar ship and they lasered it open and entered together. They pulled it apart. Well, it's not exactly that. But.
It's much, much more complicated than that. And I say that in an excited way. In other words, if it was just lasers breaking stuff apart, we kind of know how that works. Now, the problem with these viruses is that it's literally shape-shifting, right? I mean, we're taught in Bio 1 classes, everyone at some point in high school that, proteins fold into these native shapes, and then it's because of their shape that they carry out their function. And, that's great. But what it doesn't really tell us is the fact that proteins are these incredible shapeshifters, and that if you don't understand how they change shape, you basically can't understand their function. So it's not just about seeing what they look like. It's about trying to understand, again, this kind of choreography. And in the case of these viral proteins, that choreography is really complicated. It's quite beautiful. I mean, I hate to use that word in the midst of a pandemic, but from the scientific point of view, it is quite a beautiful process. It's quite amazing that something like this has evolved.
You mentioned earlier about random walk and randomness. You, more than many people, see nature and how it acts and how complicated it is. Do you still believe that a lot of it is driven by randomness, or that life harnesses stochasticity or randomness to do work? I mean, how has this changed your belief in, for instance, what constitutes life, or maybe the origins of life, or what the current dogma says about how biology acts?
Yeah, it hasn't changed at all. It's just kind of deepened the foundation of I would say, an earlier sense of how it works. In other words, I think our challenge as humans trying to understand these processes, again, is that they're happening on both time and spatial scales that are completely outside of our intuition. So a typical example, and I think one that's not that uncommon, is if you think about how we experience, just take gravity, right? So you have an apple on the table, you bump the apple, it falls off the table, right? We have that experience ever since, the age of 1 or less. We see this, we intuit it, we know what's going to happen. The apple never jumps up from the floor back onto the table, right? But for example, take gravity. Gravity is almost completely meaningless. It's not meaningless. It's just, it has very, very little impact at the molecular scale for, let's take that same hormone traveling around and binding to a cell, or take a virus. Because the forces of the fluid around it are much stronger than gravity. So for us, if you were a molecule swimming through water at the molecular scale, you wouldn't experience it as the water that we know. You would experience it as molasses, or much, much thicker than molasses. So I'm giving that example just because, again, the forces that we intuit at the macroscopic scale are not applicable to what molecules are experiencing on the molecular scale. Not only that, but they're happening much, much faster.
Yeah, I've heard that from people studying nano machines or micro machines. Right.
And I think the problem with that is that students, I mean, all of us, but we see it and we measure it, especially in students, there are all kinds of interesting misconceptions that are naturally kind of seeded when you show an animation like the one I mentioned earlier, like that version 1, right, where the molecule kind of lands gracefully on its receptor as if both of them knew what they were doing, right? That's the problem, is that's been shown so often. There's actually a follow-up study, again, with collaborators up at the University of Toronto, where we found, and this is now testing over 1,000 undergraduate students in biology, that over 83% of those students believe that molecules know where they're going. That's a big problem because fundamentally it means that you don't really understand what are the forces that are driving the system at that scale. That the molecules have no clue where they are, who they are, where they're going. The only thing that determines whether that hormone is going to bind its receptor is if there's enough attempts, random attempts, if that random walk takes it in the vicinity of the receptor, and even after bumping into it a whole bunch of times unproductively, if it happens to bump into it at the right time in a complementary way, in other words, kind of the lock and key analogy, then it will bind. And that seems like an incredibly inefficient process. But the reality is that at the speeds at which things are happening down there, it's perfectly plausible and likely, and it's in fact how things are happening. So,
but then again, there's questions like this. If you were able to model a cell, even a really simple one, like really model it, where's the life in it? What makes a cell alive?
Well, now, okay,
I mean,
good
luck figuring that out.
Now that's a great question, but it's you've taken us in a completely different direction, which is
I know. We don't have to go there, but I'm just saying, like,
it's
or
a great
even,
one. No, it's a great one.
well, I'll tell you even something else. Like, what would it take to be able to at least, I'll just say, semi-accurately model a cell in VR, where you could be in it and walk around it in VR and see most of the structures that are there and see things going on. I mean, it just seems like unbelievably complicated.
Yeah, but that's the problem right there that we were talking about before. If you were to put an infinitely, a tiny nano camera in the middle of a cell, you wouldn't see a thing, because the cell is completely packed. There is no empty space in a cell. So there's no, all the molecular animations you've probably seen are these large vistas, really beautiful, complex, usually if the, if the animator's done his or her job. And there might even be accurate proteins floating around and everything. But the reality is that you've had to remove 90% of the stuff that's in there. Otherwise, it would immediately clog the front of the camera. So
I know, but there's still a huge gap between what I've seen and what's real. I mean, you can certainly get closer to it.
There are things out there. I mean, especially in the past, I would say 5 years or so, there are definitely, including such kind of VR, tour through the cell type of things. Those are definitely coming out. But again, I think just to be clear, there's a difference between someone building this environment for you as a kind of learning experience. And I think what you were kind of the way you were saying things a few minutes ago, which is more along the lines of, can we model a cell in a predictive way? In other words, can you create a model of a cell for me such that given these changing environmental variables, something, increased temperature, give it sugar, whatever it is you're doing to it, you can predict how it will respond to it. That's a completely different, question. And both are certainly interesting challenges. But
okay.
Yeah.
You see biology, I guess, I don't know, probably in a more real frame than a lot of people, because you think about it so much and you have to model it. What's your overall gestalt when you look at biological processes? Are they messy? Do they have hidden order? I mean, I guess they do, obviously, but like, what do you feel when you look at systems where they're closest as possible to reality?
Yeah, gosh, that's so many different answers. I think the first one that comes to mind is, again, in the realm of, or in thinking about topics that we don't, that typically most of us don't appreciate, and that I think kind of blow my mind every time I'm reminded of it. Most of what we're seeing around us, and it doesn't matter what area of biology, whether you're looking at the molecular or an albatross gliding through the sky, whatever it is, we're basically looking at something that has been through millions of trial and error iterations. I mean, nature's the great engineer, right? And I think part of this, the reason I'm thinking about this, or it's on my mind recently, we're in the midst of, we're very lucky to be collaborating with a great and fantastic guy, Eric Shivian, on a book that looks at biologically inspired design and engineering as a solution for sustainable engineering practices. So a neighboring field, or what people will refer to often, is this idea of biomimicry, right? Looking to nature to uncover concepts that we might use. And so it's not just the idea of biomimicry, but in this case, we're arguing that it's biomimicry for sustainable engineering, and biodiversity preservation is kind of the key theme. But to get back to your question, I think one of the things that amazes me is that when we look at these systems, whether it's SARS-CoV-2 binding the surface of a cell, or again, a bird flying through the sky, we're looking at natural systems that have been really under such evolutionary selective pressure. All the failed experiments are gone. They didn't survive. So we're seeing machinery that is incredibly finely tuned. You always have to be careful with the language that we use, because then we immediately go into kind of the language that implies careful design, forward design. There's no forward design. There's just incredible variation followed by tough selection. And if you do that enough times and you iterate on it enough times, you wind up seeing the kinds of incredible molecular and other machines that we observe. And so I think that's a perspective that is always astonishing to me, that we are looking at designs that seem so finely tuned, so perfect for what they're meant to accomplish. And it's the power of selection. It's just that we have a hard time with our human brains imagining the number of iterations and the number of selective cycles that these things have gone through, in some cases for literally hundreds of millions of years.
Okay. Well, so, I mean, we're kind of at the end. What are some resources? I hope you've got tons of movies and pictures that we could add to the show notes, but, what resources do you have? Where can people go to see the most mind-blowing, cool animations based on their interest?
Sure. Well, obviously my first thought comes to the resources we've built. That's not to say that they're the only ones by any stretch of the imagination, but those are the ones I'll allow myself to recommend first. So my company Digizyme, of course, our website and our portfolio shows some of what we've created over the years. It's now been 20 years. But, we didn't talk about this too much today, but another important part of doing this work is to have the proper tools, to have the right software. Historically, the billions of dollars that has been invested in the entertainment industry, the Hollywood industry, has yielded really powerful software. The type of software used to make, all the Pixar movies, Lord of the Rings, Star Wars, all of those. So that's great software. The problem with that software is that it doesn't know anything kind of intrinsically about molecules or proteins or living matter, right? So a molecule in that software is the same as a spaceship or a chair or anything else. So what we've wanted to do and have done over the past 10 plus years is to create our own software that lives inside the larger kind of Hollywood software, so that we're able to leverage the strength, the power of the Hollywood software, but bring in the scientific accuracy of what we need from these data sets. So our software called Molecular Maya is basically a series of plugins for Maya, which is kind of an industry standard in 3D animation and VR and other fields. So anyway, part of our effort has been to create tools that not only we use, but also other people in our field. And then the last thing, to answer your resources question, I have always kind of really been interested and loved to teach and develop resources for the community. So over the past few years, we kind of relaunched a site that we had created earlier. Now the portal is called Clarify, CLARAFI.com. And the purpose of the site, it not only hosts our software, Molecular Maya, but it's a site that lets you browse in the showcase a number of interesting animations that we've selected and curated. So they're not just our work, they're the work of many people. And also there's a series of online courses and training that allow people who are interested in scientific visualization to enter the field, to learn how to use the software and to create their own work. So so there's some of the resources available.
Well, very good, Gail. This is awesome. I love this kind of visualization stuff. So thanks for the resources and everything. And it's really cool what you do. I mean, you have like a super amazing job. So thank you for coming on the podcast.
Yeah, my pleasure. Thanks for having me.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.